Liberty Galvin completed her Masters in International Agricultural Development at UC Davis in June of 2017. Her research was coordinated in partnership with the Njala University Extension Services in Sierra Leone, West Africa, and funded by a USAID Research Innovation Fellowship for Agriculture grant. The research objective for her master's thesis was to identify and evaluate options for increasing and maintaining knowledge transfer between extension agents and local growers within a post-Ebola setting. Currently, Liberty is a PhD student in the Horticulture and Agronomy graduate group at UC Davis, studying under Professor Qasim Al-Khatib in the Weed Science program. Sorry if I messed that up.
No, it's fine.
Liberty's dissertation research entails assisting extension specialists with developing integrated management practices for reducing pervasiveness of weedy rice in Californian rice crops. During August of 2018, Liberty had the opportunity to travel to the International Rice Research Institute in Los Baños, the Philippines, to attend the Rice Research to Production course. While there, Liberty, Shuvo, and myself became acquainted. In 2019, Liberty received the Marlon D. Brandon Scholarship from the California Rice Research Board. Her research is funded by the California Rice Research Board Endowment and the Justro Research Scholarship. I now request Liberty to please share her screen and begin your presentation. Thank you.
Thank you. Let me get this on here. Excuse me for a moment. Okay, can you see everything okay? All right, so thanks Jacinta for that introduction. Like she said, my name is Liberty. I'm coming into my 4th year of my PhD at the University of California, Davis in the United States. Once I complete my PhD, it will be in horticulture and agronomy. And I currently study under, Sarah and I are colleagues, and we study under Qasim Al-Khatib. Today, I am going to be presenting the outline, concepts, and preliminary data from the last 3 years of my PhD program. If you have a question about anything specific, I have put slide numbers in the bottom right-hand corner just so that you have a reference. It's also very important for the audience to know my talk and Sarah's talk are going to be very different from what you've heard thus far because the Al-Khatib lab is a very applied group. Our lab is funded, like Jacinta said, by the California rice growers, and we as students and staff are expected to answer research questions that are brought to us by growers. So my research was mostly conducted in a field setting, and I work directly with other field scientists and extension agents, so the outcomes of my PhD research will be used by my superior colleagues to improve management strategies for weedy rice in California. So with that in mind, oh, I'm not going to the next slide here. Uh-oh. There we go. Okay, here's the agenda for my talk. So I will give you all some background information about important agronomic practices in California rice. I'll then introduce weedy rice and what options we have to manage this pest. Then I will introduce the dissertation objectives with a conceptual diagram to help the audience understand how each aim or chapter fits together as a whole. Then I will go through each aim or chapter and discuss the methodology, preliminary data, and conclusions, as well as the research I plan to do in order to complete each chapter and consequently my PhD. So to begin, I would like to quickly familiarize the audience with California rice because I'm sure it's very different from probably all of the regions where the audience members are from. So in California, we direct seed our fields using airplanes, as you see in the video here on the right. We make aerial applications of some herbicides and pesticides. Our fields are flooded all year round with a single crop during the warm months from May to October, and then they stay flooded during the cooler months to provide habitat for migratory birds. A little fun fact, certain organizations like to refer to California rice as the environmental crop because of the ecosystem services provided for migratory birds. Now, with all that said, the lack of crop rotation, as well as a few other factors that I'll mention in a few slides, are credited with the recent presence of Weedy rice in California. So many of you may not know what may not work in rice, so let me explain to you what Weedy rice is. Also, if you have a moment, just try to ponder this question. Which one of these are cultivated and which one of these are weedy? I'm not going to tell you. You just have to think about it for a while. So weedy rice is a conspecific with cultivated rice, which means it is the same genus and species as cultivated rice. So before I tell you more, here's a quick map of California so that you get kind of an idea of the infestation. In 2018, there was about 5,000 hectares out of this 200,000 hectares infested, and all of the rice growing counties in California have been infested with weedy rice. California weedy rice has characteristics that are similar to other weedy rice that you can find in other parts of the world. They have an off-colored hull. Many of them have awns. Most have some level of dormancy, high shattering, as well as early maturity. In addition to this, my colleagues here in California have demonstrated through a phylogenetic study that California weedy rice is the result of hybridization between wild weedy and cultivated varieties from across the US. So essentially, the conclusion from that particular paper was that the weedy rice that we have here in California was brought here through contaminated seed. It likely did not originate here in California. Now, I often get asked by people, why do we care about weedy rice? Why don't we just eat it? So to answer the question, the countries where most California rice is exported to have very strict import standards, so the off-color variety is not acceptable. Additionally, many of you may not know, but shattering is when the grain easily comes off of the stalk. So if a rice plant has a high shattering rate, it's very difficult to harvest. When the harvester goes over it, all the grain will fall to the ground, and the grain is what we eat. So it's not worth it for growers to try to grow weedy rice. Now, considering all these factors, there are scenarios that have been reported in California where weedy rice has caused up to 50% yield losses in California. Before I move on, I would like to note that due to low infestation rates, my dissertation only incorporates types 1, 2, 3, and 5, and excludes types 4 and type 6. I do use M206 consistently throughout my dissertation as a control group for comparison. M206 is a medium grain, medium maturity variety, and it's very common. Many growers grow this in their fields. So moving on, many of you may be wondering, okay, so you have this weedy rice problem. What are the current options for growers to decrease this pest in their fields? So it's important to note that compared to the other rice growing regions of the United States, here in California, we are constrained by a minimal number of herbicides, and we do not have any herbicide tolerant varieties, which are common in the southern US. So this means that most of our current management strategies are cultural practices. So growers have the option, as they should, to clean their equipment. They are now required by law to purchase certified seed. Regions of the United States, growers can choose to burn their straw, but they have to get a permit, and they are only permitted to burn 10% of their fields. I know it's very different in India from what I've heard, so only 10% are able to burn. You can also do no fall tillage, but that increases your nitrogen requirements in the spring, so many growers do not take that option. Organic growers, and I'm sorry, I mean to say this, here in California we say growers, but growers and farmers are the same people. We're growing, we're farming, producers, all the same. So organic farmers can also choose to do a deep flood, which is typically about 30 centimeters, but this is not really a common practice for most farmers. And crop rotation is an option in some counties, but the majorities of counties have very heavy clay soils, and so crop rotating is not an option. Now, last but not least, the stale seedbed methodology is the most promising option. And my research is actually meant to contribute to the current efforts to develop the stale seedbed methodology, which I'll explain in this next slide. So when growers prepare their field in the spring, they have to drain the field and ensure that it is totally dry so that the cultivator can go through the field without getting stuck in the mud. After this process, growers have the option, weather permitting, to implement the stale seedbed methodology. So to do that, they prepare their field, like I said. They apply some amount of irrigation. This irrigation encourages the weeds to grow. They'll wait for the weeds to appear to some percentage. Then they implement their control strategy. This typically involves drying the field again and applying herbicides, or in the case of organic farmers, a very shallow cultivation. And after this control strategy is implemented, it's assumed that most weeds within the top 5 centimeters have been totally destroyed. So that kind of means that your crop can grow uninhibited by weeds. So for the purposes of my dissertation, I'm asking, when along this time scale do these weeds happen? Like, when are they appearing? When are they emerging in the field? We have to ask this question because the timing of implementation of the control strategy is dependent on this knowledge. So if we were to implement our control strategy at this time point, we would only be catching about 33% of our weeds, versus here where we're catching the majority of our weeds. So to kind of narrow this a little bit further for the audience, our main focus, my main focus for my dissertation is I'm trying to identify what amount of irrigation and what range of temperature, which I'll translate temperature and time in a few slides, what amount of irrigation and what temperature affect germination, which is happening here, and what burial depth and irrigation flood level are influencing emergence. Okay, so now you have kind of a picture of the questions I'm trying to ask for my dissertation. So the overarching objective for my research is to create thermal time models using ecological indicators in order to identify the emergence timing of weedy rice under field conditions. Now, this objective is broken into 3 chapters, which is a common expectation for many applied PhDs in the US, and I believe it's common in other countries as well. So the remainder of my presentation will be spent talking about each of these chapters here. And so I'm not going to discuss them much more here. However, from an applied modeling perspective, I want to show you how all 3 of these chapters fit together. So before I was showing you time on the x-axis, but time can be measured in Celsius days, which is an accumulation of heat across a temporal scale. And I also want to point out that Celsius days is the unit for thermal time, but thermal time, growing degree days, and Celsius days are all interchangeable terms. So we have thermal time here. Based on 2018 data that I recorded, roughly 18 Celsius days occurred within 24 hours, and about 120 Celsius days happened in a week. So if you look at this particular scale and you use it as a reference, we would say, like, here at this point is roughly 1 week. Here is roughly 2 weeks. Okay, so first we have to determine how much thermal time is required for seeds to reach some theoretical maximum germination percentage, which is what we're showing here, under various temperature and water potential scenarios. This is first curve. Okay, then we conducted a separate experiment to determine how much thermal time is required for pre-germinated seeds to reach some theoretical maximum emergence percentage under various burial and flooding depths. These models are theoretical because they're conducted under controlled environments. We conduct lab experiments because we can't possibly test all temperature, flood, soil, weather conditions, etc. in the field. However, the objective here, because we are operating in an applied setting, the objective is to create something that can be used by growers. So we create these theoretical models, and then the final model is adapted from field observations where the environment has a greater influence over seed development, as well as coleoptile elongation, timing, and quantity. So what I'm showing here in this slide is what may actually happen in the field. In the lab, we can reach maximums because we set all the conditions to optimal. However, in the field, all the seeds may not germinate. Of those seeds that germinate, not all of these seeds will emerge. So we are left with an actual picture of the plants that have emerged from the soil. At the end, we will compare our lab results, this one, to the field data to determine how different our lab results are from the field. And I will revisit this concept in chapter 3. So from an applied perspective, once a farmer or an extension agent or an advisor is familiar with how much heat or thermal time is required for these seeds to germinate and emerge, they can better predict the timing of when to apply their scaled seed methodology or maybe a different control strategy that will be developed in the future. Okay, so now you have kind of the background and the objectives, and I'm going to start discussing the framework of each chapter. So for my first chapter, the objective is to quantify hydrothermal time thresholds for California Weedy rice accessions to predict germination under variable temperature and moisture regimes. So to achieve this objective, we have to answer 2 questions. First, what is the range of temperatures within which Weedy rice seed germination will occur? So here's an example of how Weedy rice seed from Iran, here's the reference here, have behaved under a range of temperatures. But I would like to note that most seeds, regardless of whether they're weeds or crops, behave in this particular pattern. And this is pretty common in the literature. In this graphic, we have our base temperature here, our optimal temperature, and our ceiling temperature. These are also sometimes referred to as cardinal temperatures. Our base temperature is where physiological processes initiate within the seed. Most agronomic crops, including rice, have a base temperature of about 10 degrees Celsius. Optimal is when conditions are just right for germination, which is somewhere around 30 degrees Celsius. And the ceiling temperature is the warmest possible temperature after which a seed will start to decay from the heat. The second question is how much water is required to maximize seed germination. So to answer that question, we are analyzing water potential effects on germination. So water potential quantifies the tendency of water to move from one area to another due to osmosis or gravity. And water potential can be used as a reference for soil water availability for plant uptake, and in this case, uptake into the seeds. So field saturation here is when the field is fully flooded and there is standing water on the surface, which means that all the micro and macro pores in the soil are full of water and no drainage is occurring. As water potential decreases or becomes more negative, less water is available for plants and physiological processes within the seeds are reduced. So when you have low water potential, you have limited water available. So this water potential graphic is adapted from an article in the Journal of Agronomy, which documented germination responses of Pakistani cultivated rice varieties to various water potentials. So based on their experiment, Weedy rice seed germination is likely to occur between 0 and -8 megapascals. And our assumption is that because California rice fields are flooded year-round, we assume that Weedy rice has developed an adaptation to flooding. This is also something that I've seen in some preliminary greenhouse experiments. So because of that, we assume that Weedy rice will perform best when the field is wet or less negative compared to when the field is more dry or more negative. So to pull all these factors together, we conducted an experiment with M206, our control group, as well as Weedy rice types 1, 2, 3, and 5 across this range of temperature and water potentials, like I showed you in the last slide. We created a complete block factorial design with 4 replicates per treatment combination, and we replicated this in time. Cumulative germination was recorded daily for 14 to 28 days, depending on how quickly these seeds germinated. So next, I want to show you some of the preliminary results, which are pretty interesting. So on this slide, I'm showing you the results for base temperature. We're trying to answer the question, when does germination begin based on temperature. So here what we're seeing is we have days on the x-axis. And remember, because we're recording temperature, at any point we can turn this into a thermal time temporal scale. So we have days on the x-axis, we have germination only up to 50% on the y-axis, and each colored bar represents a different Wheaty type, with our purple being our M206 control. Okay, so there's two important things to note here. Number one, type 2 and type 5, which are this orange bar and the blue bar here, have some small germination percentage at some point within this 28 days. But types 1 and type 3, which are the yellow and green, do not have any germination. What this infers is that physiological processes are being initiated for types 2 and type 5, but they are not being initiated for types 1 and type 3 at 10 degrees Celsius. We're not really looking at M206, it's just used as a comparison, but it is very interesting that M206 had really good germination at 10 degrees Celsius. Because 10C is the base temperature, we would expect to see something like this. However, many California varieties are bred for cold tolerance because we will sometimes have colder springs. We also get a lot of our floodwaters from snow melt from the mountains. So this isn't totally shocking. Okay, the second important thing to note in this graphic is the amount of time it took to reach germination. So this experiment went to 28 days. And it took types 2 and type 5 between 28 and 23 days to initiate germination at all before these seeds started to mold. Okay, so once we got these results, we wanted to continue to explore the base temperature, because from this graphic, we don't know what the base temperature for type 1 and type 3 are. So here on the right side of your screen, this graphic is a little bit different from the one on the left, so let me set it up. On the x-axis, we have water potential becoming more negative with a full field saturation here, fully flooded field. And on the y-axis, we have total germination up to 100%. This is also at 15 degrees Celsius compared to the 10 degrees Celsius in the previous graphic. So here we're looking at a full range of water potentials because there was germination at multiple temperatures and water potential combinations. Compared to here, I only showed you results at 0 megapascals because there was no germination at any other water potential. So 3 important things in this graphic. Types 1 and type 3, the yellow and the green, they did have some germination at water potentials at or less negative than negative 0.4 megapascals. This infers that germination base temperature for types 1 and type 3 is likely closer to 15 Celsius than 10. And at 15C, these groups are not as greatly affected by water potential compared to what we saw at the colder temperature. The second thing here is that we are noticing general downward trends of germination as water potential becomes more negative. And some of these types, like type 5, which is this blue bar, which had 100% germination at full field saturation and 5% germination at negative 0.8 megapascals, is obviously very sensitive to water potential compared to these other Wheaty types that are not experiencing as big of a change. Okay, the third thing you'll notice here is it only took 21 days instead of 28 days to reach a max germination. So what we are seeing is that as the temperature gets closer to optimum, we are accumulating more thermal time for each 24-hour period. Okay, so moving on. Next, we want to look at what's happening at the optimal temperature here. So this is a similar graphic. We have our days on the x-axis, which can be converted into thermal time, and we have our percent germination on the y-axis up to 100%. I chose to show you the results from sort of an intermediate water potential, negative 0.4, to demonstrate that the field doesn't have to be fully saturated for a long period of time in order for seeds to germinate, as long as the temperature is close to optimum. So here, it only took the seeds, including the control, about 8 days to reach max germination, where if you remember from the previous, it was 21 days, 28 days, and at optimum it's only it's only 1 week. So kind of some preliminary conclusions. There are obvious differences between the Weedy rice germination patterns. When temperature